lecture today. I think you are aware now that we have plasma membranes of excitable cells. They can have what we are calling resting membrane potential. And we have talked about changes in the membrane potential to a less negative or to a more negative potential upon changing the activity of certain channels. For example, if you have activated more potassium channels, you are recording a more negative potential at that membrane than the resting membrane potential, and we have called that as hyperpolarization. Also, we can have what we are calling depolarization by increasing, for example, activity of sodium channels at that membrane. So this is one of the properties of the plasma membranes of excitable cells. They can have fixed resting membrane potential, but that resting membrane potential can change upon changing activity of channels, and we can develop what we are calling action potentials at these plasma membranes. So this is the second property for the plasma membranes of excitable cells. They can develop action potentials because they are equipped with what we are calling voltage-gated sodium channels, voltage-gated potassium channels, and by the activation of these channels, we can get this configuration of the changes in the membrane potential versus time. So what you are recording changes versus time. In x-axis is time, and in y-axis here is the change in the potential. So this action potential that is developed is having a certain or a specific time and is having many phases. According to this slide, so the phase 1 is representing resting membrane potential. We said we can have depolarization of that membrane to reach a point which is called threshold. So we are getting the phase 4. Some they are calling that change as sub-threshold potentials that can be developed, or graded potentials, whatever you would like to call it is okay. So once you have reached the threshold now, you have activated almost all voltage-gated sodium channels. So by activation of all voltage-gated sodium channels, you are getting that phase, which is the phase 3. And the potential actually is trying to reach the equilibrium potential for sodium, but is not reaching that because at the tip of that change here, at the maximum, we have other changes that are taking place. So at the end of 3, where's the 3? Here, okay, at the end of 3, we are getting inactivation for the sodium channels, and also we are getting more activity for potassium channels. And by that, we are getting this phase of action potential, which is called phase 4. So now you are aware that we have what we are calling threshold, and what we mean by that threshold. You have very fast and high reflection up of this change in the membrane potential and becoming positive, okay? What we mean by becoming positive inside with regard to outside. So that phase of action potential, some they are calling it also depolarization, but some others, they are calling it as the firing stage of action potential, okay? That's the firing stage of action potential, which is the phase 3. Then once you have reached the tip and activated more potassium channels and activated sodium channels, which have been activated at the threshold, you are getting that phase, which is phase 4, and it is called repolarization. Why we are calling it repolarization? Now, the resting membrane potential. So these cells, which are the excitable cells. Some they are calling them as polarized cells. So they are establishing resting membrane potential negative inside with regard to outside, which is meaning that these cells are polarized cells. So that phase is returning back to polarization state. And because of that, we are calling it as repolarization. Some of them, some literature are calling that phase, which is phase 4, as falling phase of action potential, falling, returning back to the resting potential. Now, you can record also another wave here, which is phase 5, which is a more negative potential than the resting membrane potential. And that phase is called after hyperpolarization phase. In some literature, they are calling it as the positive after potential. Why we are calling it positive after potential, and that wave is more negative than the action, the resting potential. Why we are calling it after hyperpolarization phase, and even though we have no hyperpolarization before. Anybody can have an idea why we are calling it in that way? Okay.
دكتور ايش هي بالضبط؟
ايش
هي بالضبط البوزيتيف
the wave 5 هذه الفيز 5 the phase 5 it is called positive after potential. Some they are calling it as the after hyperpolarization phase, the whole phase, that phase 5. Okay. Why we are after hyperpolarization? We have no hyperpolarization.
دكتور عشان ممكن اتجاه التيار
Okay.
البوتاسيوم بيطلع
because after it reached the action potential.
Okay. To answer this question, actually, for the first recordings of this whole action potential, it was recorded as changes outside with regard to inside. What that means, if you are looking to all recording in a mirror image, so you have started from the positive, then you have that wave down, which is this phase, then you have the whole action potential down, then you will have a wave which is becoming positive here, okay, more positive than the resting membrane potential. That's the first recordings for the action potential. So it is recorded by recording changes outside with regard to inside. But in this recording here, we are recording changes inside with regard to outside. Now imagine that you have a record with a mirror image. So you will get depolarization down, then the action potential, then that wave, which is positive after the potential, okay? And because of that, some they have called it as the positive after potential. And they are recording that wave in that positive direction, more positive in this way, outside with regard to inside. It was called hyperpolarization, okay? So simply that wave has kept after hyperpolarization. So the reflection down was called hyperpolarization, and that wave was after this wave, the action potential, which was called as hyperpolarization wave. And simply that wave 5 or the phase 5 has kept the old name. The old name was the positive after potential, and some they have called it as after hyperpolarization wave. Simply in that way. Why we are having that phase?
دكتور لأنه قنوات البوتاسيوم بتسكر أبطأ من
because we have still higher activity for potassium channels, so we are trying to reach the minus 94, which is the equilibrium potential for potassium. But at that phase, during that phase, we are getting inactivation also of these potassium channels. And by inactivation of these potassium channels, then we are returning back to the resting potential, which is the phase 1. So these are the phases of action potentials. Some from the resting, we can have what we are calling graded potential. Some they are calling it as depolarization. Then once you have reached the threshold, that fast depolarization is called firing stage in most of literature. Then we have repolarization wave, okay, or falling phase of action potential. Then we can record that phase, which is phase 5, as a wave which is called positive after potential, and in some literature it is called the after hyperpolarization wave. Then we are returning back to the resting potential. Okay, so this happens by changing activity of channels, as you see. So you can relate each phases to the activity of each of type of channels, sodium channels and potassium channels and so on. Okay, so again, these are the phases: 1, 2, reaching the threshold, 3, 4, 5, then returning back to phase 1. Okay, as you see here. And everything is related to the activity of channels. تفضل يا بنتي.
يعني بس بدي أسأل إنه ليش سميناها positive after potential؟ عشان في positive feedback للقنوات
No, I told you that's the old name for this phase. First recordings for the action potentials was upside down, recording the changes outside with regard to inside. Okay, and because that wave was positive after the action potential, they have called it positive after potential. Simply in that way. وضحت؟
تمام، شكرا دكتور.
اوكي. So تفضلي.
نوضح قنوات البوتاسيوم.
ايش؟
الفايرنج ستيج ثالث مرحلة فيهم.
Firing
قنوات البوتاسيوم.
That firing stage, we have no role for calcium, only sodium. We have high activity for sodium channel. Actually, that is seen here. Okay, so once you have reached the threshold at that point, to say, you have activated, we have very fast activation for sodium channels. You are starting activating potassium channels also. But once you have reached the tip of action potential here, like this one, what happens to the activity of sodium channels? These are inactivated. So the conductance, as you see here, for sodium is decreasing fast. But still we are activating potassium channels, as you see. So the conductance for potassium is still increasing. And the highest conductance, as you see here in this slide, for potassium is during the falling phase of action potential. So the first recording up is reflecting the action potential. Down is reflecting conductances for ions. In red, what you are seeing is the conductance for sodium. In blue, what you are seeing is the changes in the conductance for potassium. What you are seeing here in light green line is representing what the sum of conductances for sodium and potassium. Okay, so by summation of these conductances, you know, they are, both of them, they are positively charged particles, but sodium is moving from outside to inside, while potassium is moving from inside to outside. So at the start, we have very high conductance for sodium, but lower conductance for potassium. And because of that, the sum of the two conductances is favoring the high reflection of the potential up here toward the plus 61 millivolts. Now we have this phase, which is above zero, okay? That phase, which is above zero here. What we are calling that phase, actually, it is having a name. It is a reversal, overshoot, that's true. It's a reversal of polarization phase. So what happens during that phase, the membrane becoming positive inside with regard to outside, and some they are calling that phase as the overshot of the action potential. Okay, that's the overshot. So that period of time from this point to that point is the overshot. What do you mean by that overshot? When we have the potential becoming positive inside with regard to outside. So this is the relations that we are having with regard to conductances of the membranes, to potassium, to sodium, the sum of conductances, and here their relation with the changes in the potential to develop the whole action potential that we are having. So now you are aware of these types of recording. تفضل.
شو كان هاي الفاير شوت وين كانت بالضبط؟ لأن أنت لما أشرت ما تحرك الماوس معك.
That firing stage.
Firing state.
During the firing stage, you will have highest activity for sodium channels. So you are getting highest conductance for sodium, and because of that, you are getting this firing stage. Now, I think you have heard about the none or all principle. What do you mean by none or all principle?
When all the channels of potassium opens.
إنه إذا they reach the threshold بصير في عنا action potential. If they don't خلص ما بيكمل.
Exactly. That means you can either develop an action potential or you can have a situation where you cannot develop action potential. In which situations we cannot develop action potential? If you have small depolarization, not reaching the threshold, okay, and that small depolarization is followed by repolarization back, and some literature they are calling these potentials which can develop and not reaching the threshold to generate the whole action potential. Some they are calling them as sub-threshold potentials. Some others they are calling them as graded potentials that can develop over these membranes. Okay, so you can have a situation.
Doctor,
you can get small depolarization without reaching the threshold, and you are getting back toward the resting potential. And because of that, I am actually preferring to call the change in the membrane potential as depolarization, and the phase 3 here to call it as firing state of the action potential. So not all depolarizations can result in generation of the whole action potential. تفضلي يا بنتي.
هلا الثريشولد هو اللي بيخلي القنوات تفتح ولما بتضل فاتحه لحد ما توصل الاكويليبريم ولا لا؟
ايش؟ اي قنوات؟
يعني مثلا الصوديوم انه بس يوصل الاكشن بوتنشال للثريشولد بيفتح القنوات.
Yes, it is trying to reach the equilibrium point for sodium, but we are not reaching that actually. يعني بعد equilibrium potential for sodium is plus 61. Okay. بس احنا ما بنصل plus 61. At any time you have the overshot here. What happens to sodium channels? You are starting inactivating these sodium channels. Once you have inactivated sodium channels and you're having higher activity for potassium channels, so you are getting fast reflection back toward the polarized state for the membrane. And we are getting in this case what we are calling phase 4 or falling phase of the action potential, or repolarization phase of the action potential. وضحت؟
اوكي.
اوكي.
دكتور ليه ما بيكمل 61؟
ايش يا بنتي؟
ليش ما
دكتور
بيكمل 61؟
ليش ما بعمل؟
ليش ما بيكمل انه لل 61؟
Because you have two events have taken place. What are these events at the tip here? You are getting
يعني شو
what?
activation للبوتاسيوم.
Exactly. You have inactivated potassium channels here. Sodium, sorry, channels. And starting to act, to get also, we are getting more potassium channels active. Once you have more potassium channels active, what happens to the conductance for potassium? Still is increasing, as you see, and you are getting the highest conductance for potassium during that falling phase of action potential. Okay, but at the tip here, you have inactivated these sodium channels. By inactivation of sodium channels and having higher activity for potassium channels, so higher conductance for potassium, you are getting that potential returning back toward the resting state, which is the repolarization phase. وضحت؟
يعني هي اللي ما بيخليها توصل للبوزيتيف 61 انه
Yes, you have events that have taken place. You have closed sodium channels, so you have inactivated these sodium channels. You have also activated more potassium channels. If you have activated more potassium channels, potassium is moving from inside to outside. So the potassium is what doing? Even at the resting state, what is doing? Doing hyperpolarization, صح ولا لا? So making that potential more negative. So we are returning back, the potential becoming negative, more negative, more negative, until reaching back the resting potential. وضحت؟ اوكي.
دكتور
تفضل.
هلا تفضل.
بدي اسالك عن الجراف تبع الكوندكتنس، هذا السلايد 34.
ايوه.
راسمين انه الصوديوم بيفتحوا بنفس الوقت، انه ببلشوا مع بعض.
Yes, both of them they are starting at the threshold. That's true. But the rate of activation for potassium channels is lower than the rate of activation for sodium channels. So what you are getting very fast activation for sodium channels, but at a slower rate you are activating potassium channels slowly. You are reaching the highest activity for potassium channels during the falling phase. That's true.
تمام.
Okay, now
دكتور الرسمة اللي فوق ايش بتمثل بالضبط؟
ايش؟
السلايد 35 الرسمة اللي فوق.
This one, that one up is reflecting the changes in the brain potential, which means it is an action potential that is developed. Below it is the sum of conductances for both ions. This one in green, light green, is the sum of conductances. You know, you have two charged, positively charged ions moving, one from outside to inside, one from inside to outside. The sum of conductances for both ions from electrical view is reflected by that green line. Okay. Now, you know the phases of action potentials. You know what we mean by threshold. You know what we mean by the overshot. You know what we mean by after hyperpolarization wave. Okay. And you know what happens during the whole configuration of this action potential. So after stimulation by any mean, you are getting depolarization. If that depolarization reaching the threshold, you are getting all action potential in that configuration. If that depolarization not reaching the threshold, you will not get recording of an action potential. So that's the none or all principle. Now, تفضلي.
دكتور هلا شو هي القناة اللي تخليني أوصل للثريشولد؟
Okay, you will understand this point when we are talking about summation of events. Okay, it is much better saying now to understand this point. Actually, are we having the same threshold for all our cells? Depends on what.
No. Not the
Are we
same.
having the same resting membrane potential first? No.
No.
We know why. Okay. Are we having the same threshold potential? Also, no. That depends on what? Concentration of channels per surface area. If you have very high number, for example, of voltage-gated sodium channels per surface area, and they are in a specific state, we'll talk about these states, okay? We will talk about these states and relating these to changes in the membrane potentials. Now, we said at the threshold what happens, okay? We are activating what types of channels?
Sodium.
Sodium channel.
At the tip here, we are inactivated these sodium channels, and because of inactivation and getting higher activity for potassium channels, we are getting repolarization, okay? So, in which state we have sodium channels when we are at the repolarization phase? Simply closed or opened?
Closed.
Closed. And once you have returned back to the resting potential, in which state they are?
Closed.
Closed. Actually, we have 3 states for sodium channels, not 2. Okay? So, we can have them in the state of closed but capable for opening. We can have them opened. We can have them closed but not capable for opening. Okay? Now, once you have reached the threshold here, okay? So what you are getting, what the state of sodium channels you are getting? Opened. Activated or yes. Once you have reached the tip, these sodium channels are transformed from opened to closed but not capable for opening. Not capable for opening. So during falling phase of action potential, we have them in the state of closed but not capable for opening. What do we mean by not capable for opening? Okay. Now, if you have changed the potential during that phase, still the potential, as you know, is like a depolarization state. Still, you have a potential higher than the threshold potential. Okay? If you have that potential a little bit higher than the threshold potential, these channels will not open, even if you have the potential is lower than the threshold potential. Okay? If you have, for example, stimulated that membrane again by 10 millivolts, for example, they will not reopen again. And because of that, we are calling that state for sodium channels as closed but not capable for opening. Now, once you have returned back toward the resting potential, even the more negative potential, which is the after hyperpolarization wave here phase, causing more transformation of these channels from closed and not capable for opening to closed and capable for opening. What we mean by capable for opening? Now, that threshold potential can cause these channels that change in the potential from -70 to say to the -55 can cause these channels to open. So they are capable for opening if you have stimulated that membrane by a threshold potential. But during that phase, which is the falling phase, if you have stimulated that membrane again, okay, by that threshold potential, you will not get them activated a second time. And because of that, we said that we are having these sodium channels in the state of closed and not capable for opening, while at the resting state we have them closed but capable for opening. During firing stage we have them opened. Okay, now because of having these three situations or states for the sodium channels, we are defining a period or periods which are called refractory periods of action potentials. What we mean by refractory periods of action potential? A refractory period, a period at which the membrane cannot develop again a new action potential. Okay? Cannot develop again a new action potential by stimulation. Now we have two phases for the refractory period. We have the absolute refractory period and we have the relative refractory period. What do we mean by the absolute refractory period? Actually, in the absolute refractory period,
زي overshot.
even though you have applied a stimulus, subthreshold or suprathreshold potential, with suprathreshold potential, very, very strong stimulus, you will not cause that membrane to depolarize again. Okay, which is absolute, never ever. Why? What's the purpose of stimulation? Simply ask yourself, what's the purpose of stimulation? Is to cause these channels to open. If you are having them opened, are you changing anything by that stimulation?
No.
Nothing. Nothing
No.
will. Now, after having these sodium channels in the state of closed but not capable for opening, so during phase 4 here, if you have applied threshold stimulus like this one, you will not cause these channels to open again. But if you have applied very strong stimulus, you may cause these channels to open again. So we are defining another period which is called relative refractory period. That's the difference between the absolute refractory period and the relative refractory period. During the absolute refractory period, you will not get a new action potential generated by stimulation. During the relative refractory period, you may get a new action potential generated if you have applied very strong stimulus. Okay, but in our body we are not having very strong stimuli to cause reactivation of these sodium channels which are in the state of closed and not capable for opening to cause them to open. Okay, we are not having that in our body. But in the lab you can do it by applying very high current, for example, to cause these channels to open. تفضل.
دكتور يعني relative refractory period لو قدرنا نرجع نسوي فيها action potential ممكن يكون مثلا السيارة العصبي أسرع.
Okay, you will understand this point is not affecting the velocity, okay, of the, you will see what are the factors, what can determine the velocity of propagation of action potentials when we are talking about propagation of action potentials. ما له علاقة refractory periods, okay, but it is having one effect by having the action potential, for example, moving along axon in one direction only. يعني في هاي الحالة إذا بتتذكروا اوكي، إذا بتتذكروا السلايدز اللي حكينا عنه هنا، مثلا هذا السلايدز Once you have these currents, okay, and you have depolarized that, reaching the threshold, getting action potential here. Then we have getting action potential here. Now, that once you have action potential later on here, for example, still you can have currents actually. That is repolarizing, for example, and that is at the overshot, that part. You will have currents, but these currents are not capable for reopening of the sodium channels again and forcing the potential or the action potential which has been generated to move in one direction only. You will understand that when we are talking about propagation of action potential and the importance of having refractory periods at these membranes or at the membranes of excitable cells. Any other question؟
دكتور
تفضلي.
دكتور هلا اذا كان الستيمولس ما كان سترونج ما بيعمل نيو اكشن بوتنشال.
It will not cause a new action potential if you have the state of sodium channels are closed and not capable for opening. Okay.
Okay, تمام.
And you have applied sub-threshold stimulus. But if you have applied supra-threshold and very strong stimulus, and that happens in the lab only, you can cause these channels which are closed and not capable for opening to reopen again. When absolute refractory period, here, what you are seeing in like in green, okay? The sodium channels are already opened. If you have applied sub-threshold stimuli or threshold stimuli or supra-threshold stimuli, you are not changing anything with regard to the activity of these channels. Okay, you are not, they are already opened. إذا كانوا هم مفتوحين. With the purpose of stimulus to open them. Okay, but they are already opened. Are you changing anything? Nothing will change. Simply that way, take the relation. Now, from which point to which point we have the refractory period start? Simply, we are starting from the threshold, okay, where we are ending with the absolute. In some literature, they are saying at the tip of action potentials. Some other literature, they are talking about also including the first third of the falling phase. And then we are continuing with what we are calling relative refractory period. Don't worry about the exact point where we are getting the termination of the absolute and the start of the relative refractory period or the termination of the relative refractory period. So as you see here, the termination, okay, so it is like once we have reached back toward the threshold, according to this literature, but you are finding other literature, maybe the termination is once you have reached back the resting potential. Don't worry about the exact point where we are getting the end of the relative refractory period or the start of the relative refractory period or the end of the absolute refractory period. Could be at the tip here, could be in some literature, they are including also the first third of the falling phase. But the start of the absolute refractory period, okay, so we have good understanding that this is starting at the threshold potential. Okay, so this is تفضل.
بدي اسالك هسه احنا بنحكي الستارت للابسولوت ريفراكتوري بيريود بيجي مع الثريشولد. هسه لو اجى كمان ستيمولس ثاني قوي انه سوبرا ثريشولد ممكن يسرع الاكتيفيشن
You will not change anything. Once you have started the absolute refractory period, you cannot change the activity of these channels. They are already opened. The purpose is to open them.
قصدي يعني بصير اوبننج اسرع بصير مثلا.
No, the same thing. Once you have reached the threshold, they are almost very fast activation for almost all sodium channels at that phase. Very fast activation.
دكتور ممكن سؤال؟
تفضل.
هل الاوفر شوت من وين تبدا؟ من الستروب ولا من
Once you have started to record positive potential inside with regard to outside, so from this point to that point, that period of time, okay, as you remember, we said the x-axis is time. That period of time is called the overshot period, okay? So that's the overshot period of time. So once you are getting the potential above zero inside with regard to outside.
Okay, thank you.
Okay, now تفضلي.
بس للتأكيد هلا بالابسولوت والريلاتيف ريفراكتوري بيريود بتكون ال NA channels inactivated.
Yes, they are closed, which means inactivated,
Yes.
but not capable for opening. What we mean by not capable, this is the third state in which we can have the sodium channel. They are not capable for opening. So not capable means that the sub-threshold potential or the threshold potential, the change in the potential with the values of the threshold potential, for example, cannot cause them to open. But in the lab, much stronger stimuli can cause them to reopen again. But that happens in the lab, not in your body. So what is important in our body is the whole refractory period, the whole.
Okay.
During action potentials in our body, we are not generating a new action potentials. That's what we mean by a refractory period. Okay.
دكتور
تفضل.
البيريودز هاي اللي هي قصدي حالات الصوديوم channels اللي في السلايد اللي فوق فيش للبوتاسيوم زيها؟
No. Okay.
بس للصوديوم.
The potassium is having two states only, either open or closed. In literature, they are not talking about more than two states for the potassium, but in literature, they are talking about three states for the sodium channel. Closed, not capable. So during the resting potential, in which state we have the sodium channels?
Closed, but can be open.
Closed.
Exactly. And during that small depolarization, still you have these sodium channels closed but capable for opening. How we are getting back transformation of sodium channels from closed and not capable to opening to closed and capable to opening? Actually, that happens by returning back to the resting potential. Once you have returned back to the resting potential, even a more negative potential, as you see, a more negative than the resting, can cause more transformation of these sodium channels from closed and not capable for opening to closed and capable for opening. So that after hyperpolarization phase could be important actually for having sodium channel transformed back toward the closed and capable for opening. Okay. وضحت؟
دكتور لو سمحت بدنا نمشي هلا.
تفضلي.
دكتور هلا المرحله تبعت الفوت ستيج تمام. هلا حاط قنوات الصوديوم بتكون كلها اناكتيف طول المرحله صح؟
Inactivated.
تمام.
You are starting inactivation at the tip here, yes.
تمام. ليش اول ثلث من المرحله هو اللي بعتبره ريفراكتوري بيريود ابسوليوت؟
ايش يا بنتي؟
ليش بعتبر بس اول ثلث من المرحله absolute refractory period?
Okay, during the absolute you have most sodium channels, not all, maybe they are all also, are opened, already opened يا بنتي. If they are opened, stimulating again, you are changing nothing. The purpose of stimulation is to open them. If already they are opened, are you changing anything in an open channels? Nothing you will change. You cannot develop or having that action potential continuing also to reach the plus 61, for example, because they are already opened and you have the highest activity for the sodium channels.
بس النص الثاني هلا ما بديش الفايرنج ستيج.
Now, once you have inactivated sodium channels, that means inactivated, but still they have another property, which is closed. Inactivated meaning closed, but not capable for opening. Not capable. That means the subthreshold or the threshold potential cannot cause them to reopen again. Cannot reopen them again. Okay? While suprathreshold potential, which happens in the lab, can cause them to open again. And because of that, we are defined the second part of the refractory period as relative refractory period. Relative, which is different from absolute. Absolute, never ever. Okay? Neither with the low threshold stimuli, lower than the threshold, or higher than or suprathreshold stimuli. But in the relative, the subthreshold stimuli cannot cause them to open, while suprathreshold stimuli may cause them to open. May. Okay? Depends. Okay.
دكتور السوبرا هاد بس المختبر انت حكيت صح؟
That happens in the lab. That's true. Okay, but in your body we are not having suprathreshold stimuli to cause them to reopen again and so on. So the most important in our body, in the living systems, is the whole refractory periods, actually. The whole refractory period. Okay, that we are having.
سؤال
تفضلي.
كنت بسأل عن الصوديوم channels متى يصيرون not capable and activated؟
ايش يا بنتي؟
كنت بسأل عن الصوديوم channels متى يصيرون not capable and activated؟
ما حكينا في أي فيز، بكل واحدة بأي فيز ممكن تصير. They are closed here in that situation before reaching the threshold. Once you have reached the threshold, they are in that phase. Once you have inactivated them, starting from the tip of action potential, you are getting them closed and not capable for opening. Once you have returned back to the resting potential, you are getting transformation back from closed and not capable for opening to closed and capable for opening. Actually, we haven't much of time. I will talk a brief about involvement of other ions in that action potential. So until now, we have seen that we have involvement of sodium and potassium only, and that happens in neural cells as excitable tissues. Also, that happens in skeletal muscle as excitable cells. But if you are looking to other tissues, you can find also other ions involved in the generation of action potentials. You will take the details with regard to the involvement of other ions. I think with Dr. Faisal later on, we'll be talking about cardiac muscle, how they are generating that action potential. So only a brief, I will let you know that here we can have also calcium ions involved in the generation of action potentials in some types of tissues. Okay? And we have another properties for the conductive tissue of the heart. What do we mean by conductive tissue of the heart? As you see in yellow, we have small amounts of tissues, okay, which are called the conductive tissue of the heart, and that tissue is displaying this type of action potentials in a repetitive way. So we have automatic generation of action potentials. So simply that automatic generation of action potentials happens because the membrane of these cells are having another property, like they have leakage for sodium. What do you mean by leakage for sodium? They have all the time a little bit higher permeability for sodium. by having higher permeability for sodium or higher conductance for sodium, what you are getting depolarization. Once you have reached the threshold here, you are getting very fast activation or very fast reflection, okay, of the action potential toward the plus 10 here millivolts, then very fast repolarization returning back to the resting here. The resting is 1 point here, but because of the leakage, you are getting again slow depolarization, then
دكتور صوت
and
كثير
so on. اوكي ايش يا بنتي؟
الدكتور صوت كثير بقطع.
والله مش مني هذا،
عندك
من
لا
الانترنت، من
ادخل
الانترنت يا
وارجع
بنتي.
ادخل.
Anyway, okay. So what we have a slow depolarization reaching the threshold and so on, generation of action potentials and so on. So we have a property at that conductive of tissue by themselves. We have automatic generation of action potentials, automatic, without any stimulations from outside. So why that happens? Because we have leakage for sodium to cause this slow depolarization, and we have other ions involved, other types of currents that we can have, which are calcium currents. That means we are activated calcium channels. Okay, at the threshold here, we are activating calcium channels actually. Okay, even from the start of depolarization here, you are starting activating the first type of calcium channels, which are called T-type, which are transient. Okay. By activation of the T-type, you are getting more depolarization. Once you are near or reaching the threshold, you are activating the second type of calcium channels, which are called long-lasting type. Okay. By activation of the long-lasting type, you are getting that fast reflection of the action potential up toward the plus, okay, the plus millivolts. You are reaching the tip here. At the tip, what happens? You are getting activation of potassium channels. By activation of potassium channels, you are getting back repolarization phase, but here it is called phase 3, which are different from the phases that you are having the action potentials at the neural tissue. So we have many types of currents that can be or during that the generation of action potential. Here you have F current. What do you mean by F current? What type of ion we have here?
فلور.
Huh?
فلور.
صوديوم.
The sodium, but they are calling them funny currents actually. Funny. The F is from funny. Okay, because it is unusual. That small depolarization by that sodium current, okay, so we are getting sodium entry from outside to inside to cause small depolarization. From, for example, -60, once you have reached -50 here, you are activating the first type of calcium channels, which are called transient type. By activation of calcium channels, this type of calcium channels, which are the T-type, you are getting more depolarization. That more depolarization results in activation of the second type of calcium channels here, which are called the L-type, long-lasting type. So what you are getting? Very fast depolarization, as you see. Reaching the tip here, what happens to the activity of calcium channel, the L and T? They are inactivated, and you are starting activation of potassium channels. You are getting the phase 3 as example. So we can have different ions involved in the generation of action potentials. You will take that when you are taking the cardiac muscle actually with Dr. Faisal, I think. Also the muscle, the cardiac muscle cells are displaying this type of action potential, which is called action potential in plateau, and we have also calcium involved in the generation of this action potentials in plateau. So what you are seeing in blue is the activity of sodium channels during the action potentials. What you are seeing in brown is the activity for the potassium channels. As you see, the highest activity for the potassium channels during this phase here of the action potential, which is called phase 3. And the highest activity for calcium channels is during the phase 2 here, which is called the plateau of action potential. So this is the variation that we can have in the action potential by having other ions that could be involved, like calcium ions here. Also here we have calcium ions involved, which has, it's like clamped the potential for longer time actually, okay, until getting the repolarization phase by having or by activation of the calcium channels here. You are getting more calcium entry from outside to inside and prevents the fast repolarization here. So keeping the potential for much longer time until getting the start of repolarization. So this is
دكتور هاي
the details actually with Dr. Faisal later on, only to remind you or to open your mind that we can have tissues. Okay, not only potassium and sodium can be involved in the generation of action potentials, but we can have tissues in which we can have calcium also. Channels are involved in the generation of action potentials. وضحت؟
دكتور الجراف تاعت البلاتو هذيك ما صلصة.
Okay. I'll stop recording.